Another type of granulocyte is agranulocyte or non-granulocyte, such as monocyte and lymphocyte. Monocyte transform to macrophage or turn into macrophage when an invading germ or bacteria enters your body and kill it. Monocyte or macrophage about 3 to 8%. Mononuclear phagocytotic system, mononuclear because all agranulocyte and non-granulocyte just have a one nuclear lobe. You can see here monocyte one nuclear lobe and lymphocyte one nuclear lobe, not more than one. Call it mononuclear phagocytotic system or mononuclear leukocyte. You can see here some specific macrophage into the human body, for example, into the liver, Kupffer cell, into the bone tissue, osteoclast, into the nervous tissue is microglia, into the lymphoid tissue, dendritic cell, and into the lung tissue is dust cell. All of these cells are macrophage or specific macrophage. Kupffer cell, osteoclast, microglia, dendritic cell, and dust cell. Another type of agranulocyte is lymphocyte. Lymphocyte help your body's immune system fight cancer and foreign virus and bacteria or antigen. Lymphocytes help your immune system remember every antigen it comes in contact with. After an encounter, some lymphocytes turn into the memory cell. Okay, memory cell, because immune system remember every antigen it comes in contact with. This is the function of lymphocyte. There are 3 types of lymphocyte. A small size, 6 to 8 micrometer. Medium size, 12 to 15 micrometer. And large size, about 18 micrometer. This is normal situation, not the same erythrocyte as anisocytosis, as a pathological condition. For lymphocyte, different size of lymphocyte is normal situation, but most of lymphocyte are a small size, about 6 to 8 micrometer. About 20 to 25% of leukocyte in whole blood tissue is lymphocyte. There are 3 different types of lymphocytes. 1, B cell, produce antibody, T cell, and natural killer cell. T cell into the thymus, B cell into the bone marrow, different type of lymphocyte, and natural killer cell. 3 type of lymphocyte into the human body in normal situation. B cell or B lymphocyte, T cell or T lymphocyte, and natural killer cell. B cell and T cell for break down the virus and bacteria need to recognize the receptor of all antigen, but natural killer cell don't need to recognize the receptor of antigen. This type of cell can kill all of the antigen into the human body without recognize the receptor located the surface of antigen. But B cell and T cell need to recognize the receptor located in the surface of antigen as a biomarker. You can see in this picture monocyte. The size of monocyte bigger than other type of leukocyte, about 18 micrometer to 20 micrometer, bigger than other type of leukocyte, granulocyte and agranulocyte. Here is lymphocyte. Mononuclear lobe or one nuclear lobe here and here. You can see here the terminology. This terminology is diapetesis. This process is movement or passage of blood cell, especially white blood cell, through intact capillary wall into the surrounding connective tissue. This process called diapetesis for all white blood cell, not red blood cell, for all white blood cell. You can see in this picture diapetesis process. Here is neutrophil. You can see here neutrophil have selectin ligands. And here blood vessel. The lumen of blood vessel here is endothelial cell as an epithelium of vessel, endothelial cell, or epithelial cell for blood vessel. You can see here endothelial cell have selectin receptor. This selectin ligand bind to the selectin receptor like this. When neutrophil by using selectin ligand bind to the selectin receptor, here is rolling. This rolling induce the neutrophil to express another type of ligand as integrin. Okay, you can see here integrin. And here all endothelial cell have receptor for integrin. In the next step, integrin ligand bind to the integrin receptor located on the surface of endothelial cell. When integrin ligand bind to the integrin receptor, this attachment is so strong. The neutrophil attachment strongly and tightly to the endothelial cell. In the next step, 5, the neutrophil movement or passage through intact capillary wall into the surrounding connective tissue. You can see here the neutrophil come out from lumen of blood vessel into the connective tissue. Okay. This process called diapetesis for all white blood cells, eosinophil, neutrophil, basophil, monocyte, and lymphocyte, for all white blood cell or leukocyte. You can see in this picture the process of diapetesis step by step. The first step, rolling, binding the selectin ligand to the selectin receptor. You can see here integrin in inactive, not active in this step. After that, activation of integrin, adhesion, integrin ligand bind to the integrin receptor located on the surface of blood, on the surface of endothelial cell, and locomotion, protrusion, and transmigration between two endothelial cells come out from blood vessel to connective tissue. Rolling, activation, adhesion, locomotion, protrusion, and transmigration. When all white blood cells through diapetesis come out from blood vessel, you can see here endothelial cell is intact, not rupture the endothelial cell. This all white blood cell or leukocyte come out between this junctional complex. Okay, this junctional complex breakdown, and white blood cell come out or run out in this area from blood vessel lumen to connective tissue. After that, this junctional complex reconstructed or reshaped. Another question, which leukocytes are capable to perform diapetesis one more? For example, 2 times, 3 times, 4 times, more than one. Okay, which one? Neutrophil, eosinophil, basophil, monocyte, and lymphocyte. Which one? One leukocyte can perform diapetesis one more than 2, 3, or 4 times, or more than 4 times. Okay. Another type of blood cell is platelet or thrombocyte. One of the smallest cell into the blood tissue is platelet or thrombocyte. The size of this blood cell is about 2 to 4 micrometer in diameter. The number of platelets about 150,000 to 450,000 in cubic millimeter. The lifespan of platelet about 10 to 14 days. This type of blood cell has a non-nucleated disc-shaped cell fragment and derived from megakaryocyte cytoplasm. You can see here red blood cell. in this blood smear. Here, lymphocyte, here, platelet, or here. This one is red blood cell, all of this. You can see here, as you know, red blood cell don't have nucleus. Here, basophil. Here is platelet. You can see the smallest cell into the blood tissue, about 2 to 4 micrometer in diameter. This is platelet, this one and this one. Or you can see in this picture transmission electron microscopy of platelet. The ultrastructure of platelet. You can see the platelet are smaller than red blood cell, around 1.5 to 4.5 micrometer in diameter. And this type of cell is biconvex discoid shaped. Biconvex discoid shaped. Platelet do not have a nucleus, the same red blood cell, but they have a mitochondria, microtubule, and actin cytoskeleton, glycogen granules, some Golgi and ribosome. Okay,